Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, and very good, good morning to all of us. First of all, let me once again welcome our distinguished speaker, to Professor Peter Ilobi from the Shenzhen International Graduate School, Tsinghua University, and also Dr. Hadi Mulya Asmara, PhD, from University of Calgary. Welcome to all of students from the Fakultas Medicine and the Fakultas Farmasi to the Kolaboratif international guest lecture initiated by the biomedical program Fakultas Medicine and the Fakultas Farmasi Universitas Indonesia. So I also would like to welcome to Prof. Muli from Fakultas Medicine, and also Dr. Apoteker Santi Punasari and Dr. Baita and Dr. Dona from Fakultas Farmasi, and to all of the students who already come and join this guest lecture. So we will have two sessions here. First, we will, and I think I haven't introduced myself. My name is Meidi. I'm a lecturer and a researcher from the Fakultas Farmasi. So we will have the first session from Prof. Lobi, which will talk about the novel therapeutics in female reproductive cancer, and then continue with Dr. Hadi Mulya Asmara from University of Calgary, who will give a talk about the from animal care to research discoveries. So before we start the first session with the Prof. Lobi, let me a little bit introduce you about the Prof. Lobi background. So currently, Prof. Lobi serves as the professor at the Shenzhen International Graduate School, Tsinghua University. He finished his Bachelor of Medical Science with distinction from the University of Queensland, Australia in 1990, and continued with the Bachelor of Medicine and Bachelor of Surgery with first class honor, University Medal for Outstanding Scholarship, the University of Queensland, Australia in 1992, and continued with the Doctor of Philosophy, Doctor in Medicine, Vetenskap, Molecular Endocrinology, Karolinska Institute, Department of Medical Nutrition, Stockholm, Sweden in 1994. And for his work experience, now he served as the chair of the breast cancer funded by the Breast Cancer Research Trust, also become the co-director of the Center for Precision Medicine and Healthcare Research, and now he served also as the professor and senior principal investigator at the Cancer Science Institute of the Singapore within the National University of Singapore, and now currently he working as the professor within the Institute of the Biopharmaceutical and Health Engineering in Tsinghua University. He already has so many publications in the reputable journal, and among those paper, maybe among those article, there was these are related to this his talk today about the metabolic plasticity in pancreatic ductal adenocarcinoma progression and response to treatment. Also, the high throughput engineered tumor organoids reveal ROC signaling as an immunotherapeutic target in triple negative breast cancer. Also, the interaction of the CYP1 and HNRMP in regulatory cell transition of breast cancer cells. So, we at least we already have some idea what his talk about. So, without further ado, maybe I will welcome Prof. Lobi to start. Thank you.
Okay, thank you for the kind introduction. It's my great pleasure to be here today. I again thank the University of Indonesia Department of Medicine with Prof. Muli for organizing my visit here. It's been really quite entertaining and also educational. Yeah? I'm very happy to be here. And also thank the Faculty of Pharmacy for their invitation to visit this beautiful campus today. So this is the Shenzhen International Graduate School where I do a lot of the work. Shenzhen is a city, of course, opposite Hong Kong, a very high-tech, forward-looking, modern city. And the work we do there focuses on the development of novel drugs or novel therapeutics for use in female reproductive cancer. Female reproductive cancer in Indonesia is actually very high. The first 3 cancers are all reproductive, are all the top cancers in women in Indonesia. So breast cancer, of course, first, followed by cervical cancer, and then ovarian cancer is number 3, which is a little different to a lot of other countries, the situation in terms of cancer incidence. I've been introduced, but just to say that in addition to the usual academic things that professors do, I also have a large amount of industry experience. We are working in drug development. We do need to commercialize our discoveries if they are to benefit patients. Unfortunately, that's the reality of society, that we do need to commercialize things to be able to bring them to patients. So I've got quite a lot of consulting experience with both large pharmaceutical and small pharmaceutical companies, and also a number of director positions or founding scientist on a number of different companies to try and bring the discoveries that we make in the laboratory to the clinic. In China, you know, we can't use WhatsApp and everything, so this is my WeChat code if anyone is interested. And otherwise, this is my email here, so please feel free to contact me. So the focus in my laboratory is really understanding cancer biology of female reproductive tumors, and hopefully by understanding the biology of the tumor, we're able to address unmet medical needs, right? Cancer is a very difficult to treat disease. The outcomes are not optional, are not optimal, and we use or we study right from how cancer develops all the way through to how cancer metastasizes. As medical and pharmacy students, you're well aware that cancer metastasis is usually the reason for mortality in cancer. We can easily cut off a breast and the patient can survive, but if the tumor metastasizes to important organs in the body, then the outcomes again are very poor. So the poor outcomes happen because of chemo resistance, right? We're not able to treat the cancer properly. It happens because of tumor heterogeneity. So a tumor cell population is very similar to a human population. We're all actually quite different. And if you put pressure on human populations, they find ways, or individuals find ways to survive. And also cancer cells find ways to survive when under pressure. And also, and I think our next speaker will touch on this, a lot of the conventional models, both the clinical models and the preclinical models, are really not very good at predicting a clinical response. And one of the companies I'm involved in China, in Hangzhou, is with a company that takes tissue from the tumor, grows it in a 3D organoid structure, and determines the drug response for individual patients. And this is starting to improve the prognosis for a number of patients. So the approach we take in our laboratory is also, I guess, quite comprehensive. We have a very lot of expertise in mechanistic insights in terms of cancer biology, but we're specifically interested in proteins that enable cancer cell survival. Cancer cells, when they're dead, they can't do anything else, right? They can't escape therapy, they can't revert to a more aggressive phenotype. Once we've identified survival pathways of interest, We then go on and design first-in-class drugs. So these are entirely new chemicals targeting new targets. This work involves a lot of omics approaches these days. It also involves a lot of artificial intelligence approaches, which Shenzhen is promoting very heavily. And then once we have these new drugs, we do a lot of preclinical work, but using clinical samples to identify very specific disease states where the use of these drugs might prolong survival or even cure cancer. But of course, as everything in science, we're constantly evolving and progressing. The rate of technology change at the moment is very, very high, and we're working very hard to try and keep up and be at the forefront of drug discovery and cancer biology. Just to declare my conflicts of interest from a legal perspective. The drug I'll be talking about today is actually licensed to one of these drug companies, so I do have to declare. So what I'm going to talk to you today about is a new target that we've been working on. It's called BAD. It is a bad protein in lots of ways. But BAD is also phosphorylated, and we target BAD phosphorylation not by inhibiting kinase activity, but by the drug will directly bind to BAD and inhibit phosphorylation. So many of you probably already know that BAD is a member of the BCL2 family. This is a family of pro-survival proteins, a very prominent role in cancer. So BAD really stands for BCL2 associated death promoter. BAD is a dual function protein. It's not simply on and off. So when it is phosphorylated, and the phosphorylation should occur on 75 and 99, it will bind to a protein called 1433. Can you guys see the laser pointer or not?
Yes.
Oh, you can. I'm actually a little colorblind, so I can't see. Okay, so it binds to a protein called 1433, and that sequesters BAD away from binding to these survival proteins like BCL2, enabling them to bind to the pro-apoptotic proteins, which enable cell survival. When BAD is dephosphorylated, it dissociates from 1433, binds to BCL2, BCLW, BCLXL, and therefore promotes apoptosis. So simply by changing the phosphorylation state of BAD, we can switch this protein in cancer from being pro-survival, which is not good, to being pro-cell death, which is good from a therapeutic point of view. BAD, of course, sits downstream of these very important pathways in cancer. The RAS-RAF MAP kinase pathway and the PI3-AKT pathway. So both of these pathways are heavily targeted by different pharmaceutical companies. Right, these are a lot of the so-called targeted therapeutics that are coming or have been coming on the market for a number of years now. The problem with these therapeutics is that they inhibit one side, and if they inhibit one side, then the other side becomes much more active, right? So the cell compensates for the inhibition. And so while the effects may be good short-term, longer term, the resistance develops and the patients generally, again, experience a poor outcome. So we targeted BAD because it sits downstream of both of these pathways, and it needs one phosphorylation input from the MAP kinase pathway and one phosphorylation input from the PI3 kinase pathway. So this is an example of why the upstream drugs don't work. This is using vemurafenib. This is the BRAF inhibitor, the BRAF V600E inhibitor, and a patient with malignant melanoma. You see it's a highly metastatic tumor spread all over the body. Not a very good outcome, but you treat with vemurafenib and actually it looks good. But the problem is, after about 6 to 18 months, the tumor recurs very, very aggressively and comes back, and the patient, of course, dies of the tumor. This resistance has actually been shown to be mediated by bad protein, bad phosphorylation, indicating that bad might actually be a very good target in oncology. So we went ahead, and this is when, from Singapore, a lot of this work, with two very smart Indian fellows that I worked together with, and they designed a drug that was able to specifically inhibit the phosphorylation of only serine 99, only one residue. And that is all that is needed, just one residue inhibited. It produces a decrease in cell viability by promoting apoptosis of cancer cells across a whole range of different cancers, but not in normal cells. So we had a drug, we went back and we asked, in what situation can we use the drug? So given that bad phosphorylation is quite high in ovarian cancer, bad phosphorylation is again further increased in ovarian cancer that is treated with different chemotherapeutics, we thought that utilizing this drug in ovarian cancer might be quite useful. The problem with ovarian cancer is once it occurs, it can be treated by the standard medical approaches. It can be treated by targeted therapy like PARP inhibitors. But what happens, a number of cells survive, and they can sit there for a while, not too long, but then they come back. Most patients will experience relapse and die. So anything that will improve the survival outcome of these patients becomes very useful. So a very competent clinical fellow in my laboratory, Dr. Jiang Si, she went ahead and using patient-derived organoid and patient-derived xenografts showed in this patient was terminal. She had peritoneal ascites from peritoneal metastasis. We isolated from cells, confirmed the genotype. Here we see a mutation in the PI3 kinase pathway. And then we treated with our drug in blue and olaparib, which is the FDA-approved PARP inhibitor from 2017. So we see our drug and the FDA drug basically work the same, but in combination. We're able to obtain, if we look at the Kaplan-Meier survival curve here, we're able to obtain what we think is a cure in about 1/3 of the animals, which was a really happy result for us. But then the skeptics in the laboratory said, 1/3, it's not really good enough. We want to also further improve the outcome. So I'll come back to the ovarian cancer situation a little bit later. But anyway, the drug worked, quite efficacious, combination therapy, a very good outcome. In P10 deficient endometrial carcinoma, she also looked. P10 is of course a negative regulator of PI3 kinase. So when PTEN is lost, and it's lost in most endometrial carcinoma, the PI3 kinase pathway increases activity and increases bad phosphorylation on serine 99. So if we use the BAD inhibitor is called MPB. Not only does it decrease serine 99 phosphorylation and drive the cell to death, but it also interferes with the way PARP inhibitors work. So PARP inhibitors work in a synthetic lethal way by increasing DNA damage, right? You get a cell that already has DNA damage, you increase it a little bit more and the cell will die. But at the same time, PARP inhibitors stimulate DNA repair through homologous recombination. And bad serine 99 also plays a role in homologous recombination. So we're able to prevent the homologous recombination, which limits the efficacy of the PARP inhibitors, and really get this synergistic in combination effect, again resulting in cell death. So in these experiments, both our drug and olaparib were quite effective, but together we achieved a 100% complete response, result we were very happy about. Also in cervical cancer, cervical squamous cell carcinoma, which is a very, very prevalent disease in South and Southeast Asia, when we use the BAD inhibitor in combination with mitomycin C as a substitute for radiation therapy, which is used in this disease, again using patient-derived organoids in collaboration with the Hong Kong University Hospital in Shenzhen, where again, in combination, able to obtain much improved responses in cervical cancer. So the drug we were working with, called MPB, it wasn't very potent. It wasn't as potent as we would like it to be, and it wasn't orally available, which is a very good topic for pharmacy students. So we went back, and Esther from Malaysia, she spent a long time trying to find an improved drug that was both more efficacious, right? So a lower IC50 value, and orally available. And so we came up with this analog called NCK. So NCK still bound to BAD. We can show that using a protease protection assay. When the small molecule binds to the protein and you treat with a protease, if the small molecule binds, the protease will not be able to degrade the protein. So we see increasing concentrations, more protein. But it also bound to the BAD interactin protein called 1433, which was probably responsible for the increase in efficacy. As you saw before, combination strategies in cancer are particularly useful and much more effective than single agent therapy. Use of single agent allows the possibility, or a higher possibility, for cell escape from the therapy, survival, resistance, and recurrence. So we took our drug, NCK, and used it in a library of oncology drugs and screened for the drugs which interacted best with inhibition of bad serine 99 phosphorylation. So Esther's interest was in triple-negative breast cancer. I just spoke with a very nice professor in pharmacy who works in triple-negative breast cancer. And this is what the results look like from the high-throughput screens. It's a lot of work, a lot of drug combinations, a lot of different drugs and use at different concentrations. But it was very nice to see that even from an objective approach, we could show that PARP inhibitors were actually very synergistic with inhibition of bad serine phosphorylation, that PI3 kinase inhibitors gives a very mechanistic basis for the drug to be able to work. But we also saw that the most synergistic combinations were actually with upstream receptor tyrosine kinase inhibitors. And the two that were the most synergistic is a drug called crizotinib, which is an ALK inhibitor used in ALK-positive lung adenocarcinoma. It's also a multi-kinase inhibitor, so it inhibits a number of other kinases such as MET. And also another drug called OSI 930, which is a KIT inhibitor. KIT is a stem cell receptor tyrosine kinase. So when we use these drugs in combination, we were able to obtain actually a complete response as a single agent therapy in 2 out of 6 animals. Crizotinib slightly improved that outcome, but combination with OSI 930, this C-KIT inhibitor, actually produced a complete response in 5 out of 6 animals. I think most of you probably know that triple-negative breast cancer is extremely difficult to treat. There are really very few treatment options, chemotherapy, standard approaches, radiation therapy. So again, use of this drug actually offers a potential cure for what is currently an incurable disease. So now I'll go back to the situation with ovarian cancer. Again, a very talented Indian fellow. I work in the international campus, so we take in lots of people from overseas, so please welcome to our campus. So he went back and he looked at why in that 2/3 of ovarian cancer PDX models did resistance developed. He used the new drug, NCK, and really to cut 5 years of his work into 1 or 2 slides, he identified by various omic techniques that a molecule called PIM1, it's also a kinase, that PIM1 was responsible for the development of resistance when you treat ovarian cancer with the PARP inhibitor and with bad serine phosphorylation. So PIM1 is well known to be a kinase involved in cancer progression, stimulating a range of different cancer functions. And it's also known to be as a rescue kinase. So if we inhibit with other drugs up here, right? Tyrosine kinase inhibitors, anti-angiogenic agents, PI3 kinase inhibitors, PIM1 becomes active and bypasses the inhibition to produce resistance. And this is really, I think, one of the most exciting results we've had from the lab in many years. So what we observed was that only after 3 rounds of treatment, we only treated for 4 days, gave a rest, and did that 3 times. After 3 rounds of treatment, and in fact you can already see after the first round, the tumor regresses and it doesn't come back when we use it in combination, right? And that's pretty obvious here, but in the Kaplan-Meier survival curve, we have 100% survival of this PDX model from a woman with end-stage chemo-resistant, radiation-resistant, targeted therapy-resistant ovarian cancer. So again, using quite detailed knowledge of signaling pathways, how survival pathways work in different female reproductive cancers, we're able to obtain a very nice outcome. At the moment, we've licensed out this drug. We are pushing it forward to try and get through toxicity and CMC toxicity studies. Small animal toxicity studies show that NCK, our drug, is extremely well tolerated, so it's a very good prospect for future clinical application. So to summarize today, we only have 20 minutes. I hope I haven't gone over time already. Bad serine 99 can be specifically inhibited with one small molecule drug without inhibiting the upstream kinase activity. And inhibition of bad serine 99 may be quite useful in some female reproductive cancers, very common in Indonesia, like triple-negative breast cancer, ovarian carcinoma, and also cervical carcinoma. I have to thank a lot of people. This is a lot of work. We have a lot of international collaboration, both in China, at University of Science and Technology of China, in India, in UK, in New Zealand, and in Singapore. And hopefully soon we can add University of Indonesia here to collaborate together. I really need to thank my two senior scientists, Vijay and Jangsi. They're really instrumental in pushing a lot of this work forward. And also these are my students and postdocs, so they also work very hard to get this done. Also the funding agencies in China, Shenzhen, offers quite good support for research-orientated enterprises. So I realize most of you are undergraduate students. I hope I've made it explainable or explicable to you. Thank you for your attention, and I believe we'll have a question and answer session after the second talk, right? So thank you very much.
Thank you very much, Prof. Lobi, for the very impressive talk. So as Prof. Lobi mentioned, for the discussion part, we will save after the second part. So please save your questions. Don't worry, we will surely have the discussion for the Q&A. So let's move on to our next speaker, Dr. Hadi Mulya Asmara, PhD BMP, and now he's working on at the University of Calgary. And let me introduce a little bit background about his educational background. He was finished a Bachelor of Science in Biological Science, Faculty of Mathematics and Natural Science, Universitas Indonesia, and then continue his Master of Science in Biomedicine, Fakultas Medicine, Universitas Indonesia, and then continue to research student in molecular biology at the Kagoshima University from 2005 until 2006, and then continue his PhD in physiology and molecular biology of neurocardiovascular, Kagoshima University from 2006 and 2010. He has professional experience. He successfully initiated, led, and managed a collaborative research project from concept to publication in international scientific journal, and also participated in many leadership training and project management by the University of Calgary and also by MIPACS Canada. He has also many publications in the related field, and one of the recent publication is about the calcium-dependent regulation of neuronal excitability is rescued fragile X syndrome by tight conjugated N-terminal fragment of fMRP. Okay, so maybe that's the introduction. So without further ado, maybe I will welcome Dr. Hadi to start his session.
Assalamualaikum warahmatullahi wabarakatuh. Thank you so much for the introduction, and I'm happy to be here again, even different building, but the same program like biomedicine program, and I acknowledge there are most of you from Faculty of Farmasi, welcome to this lecture. And I met several of my colleagues, alma mater. Thank you for attending. Today, yeah, I just want to add some information, and Prof. Muli invited me here especially to talk about animal care, because I recently joined animal health unit in University of Calgary under Vice Provost Research, University of Calgary. So I'm getting involved in animal care committee ethics for any research involving animals in University of Calgary, and most of my talk about that, and then I relate with my research field in University of Calgary as well. Yes, as we know that, like Professor Lobby mentioned, most of drug discovery starting from traditional approach, which is using animal model. And later I will talk about why we use animal. Yeah, because lifespan in animals short than human. If we use human, of course we have to wait like 70 or 80 years old or even further. But using animal is life short span, and also ethical. We cannot use human as object, like Professor Robi experiment about several different targets. Yeah, so we use animal for preclinical, optimize the dose and the effect and so on. And many other experiments, not only drug, vaccine, organ transplantation, treatment, cures, also method of surgery. We use animal as object of research. And according to CCAC is Canadian Council of Animal Care Committee. So in Canada, the use of animal is highly restricted or regulated by the government through this agency. And as the data, like recent data in 2021, 3.7 million animal has been used for research, which is 10% of Canadian population actually. Canadian human population is 37 million people, so 10% of animals has been used for research in Canada. So you can imagine how many animals been involved in that we take benefit from, and mostly fish. This is interesting. Like I was assumed that mostly rodents, yeah, rabbit or mice or rat, but mostly fish, zebra fish. And as we know, maybe in biology we use embryological development. We usually use zebra fish, and also other kind of research. We use zebra fish as well. And so I talk a little about this council, because why this is so important. They oversight all the institution in Canada that involved in using animal as in research, regardless university, private company, or whoever using animal, has to go through their approval or oversight. And they do inspection. It's not only just one time thing, but every 3 until 6 years they come to that institution and to inspect what has been implemented or what has been missed from their guidelines. So they provide guidelines for using animal in research. I just make this a bit of a discussion. Why? Why in developed country mostly they really restricted using animal in research? Any one of you want to participate, giving opinions? Why in developed country the type of regulation of using animal in research really restricted? Anyone has? Yeah, like this is, many regulation. This is in federal legislation, provincial, and this is also the government funding. In Canada, there are several government funding agencies. They won't release the fund, even the professor or the PI or the lab has been approved to get the government funding, but they won't release the funding unless the animal committee on that institution approve the protocol of research. The funding will be held until they get approval animal ethics. Why? Anyone want to give opinion? Because when I develop communication with Dr. Puspita from Faculty of Medicine, me from the Faculty of Science, Mathematical and Life Science, I got the answer actually. It's different with Canada and Indonesia. There is a huge difference on that. What do you think? Anyone want to comment?
Anyone? Don't afraid.
You can shout out loud, yeah, please.
Can I use bahasa?
Yes, sure. You can use bahasa. Sorry. Yeah.
Sebelumnya perkenalkan, nama saya Kasilda dari Fakultas Farmasi. Izin menjawab. Tadi pertanyaannya terkait kenapa di Kanada lebih
Strict.
ketat,
Yeah.
Karena kalau menurut saya mereka tuh lebih memperhatikan hewan gitu. Karena mereka merasa mungkin hewan itu sama kayak kita gitu, sama-sama makhluk hidup. Jadi lebih menghargai lah sesama makhluk hidup gitu.
Probably, do you understand? Sorry. I can translate it for you. So, as he said, in Canada they more care about living organism like animal, so they protect or they have good care of these animals. Any other comment? Answer? Yes, please.
Good. If I may answer, it's about the 3R regulation. So, if I'm not mistaken, I've heard that in like developed countries, when you use animals for experiment and stuff, you have to like replace them and you have to refine your procedures to have it like to have less suffering on the animals. If I'm not mistaken, thank you.
Yes, that's the impact of that reason. Then the committee have to ensure we apply 3R principles. I will talk about that later. But the reason behind why the regulation been in place, the regulation, the oversight will they've been implemented. No guess? No other guess? Exactly. Prof. Lobby answered it for all of us. Public pressure. So there are many NGO, non-governmental organization, overseeing what's happening behind the cages. Not only the institution, the cages of those animals. What has been done to those animals? I give you an example. Just recently, like a few months ago, there is a big news in Toronto, Canada, about using dog as research for cardiovascular, because dog has more similarity for cardiac with human. Yeah, it's big news because in Canada, dog is like family. Yeah. So, in my perspective, that news either because the public doesn't know what's going on in animal care committee, so the news outlet like national news outlet, they try to blow up the news by providing misinformation about animal in research, or yeah, there is a misprocedure, yeah, or there is something wrong in the procedure, so that news become blow up. Yeah, so this is really important because, like Prof. Bobby said, public pressure. Yeah, looking at what's going on with that. So in every institution that use animal as research, they have to put up in their website the information, including the numbers of animal has been used. Yeah, this is really important. If you check any Canadian institution, they provide those details information. So that's why every institution has institutional control of using the animals. There's in institutional level. There's University Animal Welfare Committee, and in faculty or lower level they have Animal Care Committee. Yeah, and I'm involved in this committee. So what they do, they review all the research protocol. Yeah, regularly. Like we have monthly meeting, the Animal Care Committee, but the PI or the professor, the labs, they submit their animal use protocol, and then the veterinary will pre-screening. Yeah. So the important role of veterinarians, they will pre-screen whatever harm to the animal, whatever dangers to the animal, they will give red flag to the PIs. This cannot be done in animal. For example, in cancer research, I told story about when we review the proposal. There is a cancer research using rat that they will introduce cancer cell line to the tibia of mice. Tibia of mice. You can imagine tibia of mice is like small, but they will grow up the cancer until 1.5 centimeters. This is unimaginable for rats, or for mice. So the veterinarian suddenly stop. This cannot be done unless you change. So they approve until 1 centimeter. But this also has to, again, I will talk about refinement later. They have to give analgesic or opioid to reduce the pain. Yeah. So this is not only human has ethic, but also animal. Yeah. We don't want to make the animals suffer during we introduce something that can make them suffer. Yeah. So again, like veterinarian is one of in animal committee, and scientists who use the animal, and scientists who do not use the animal. So they can give perspective what should be done or what shouldn't be done in animal research. And of course technician, and this is community representative. This is ordinary people who doesn't involve in research,
Pet
lovers. But yeah, this is kind of also something tricky because we don't want people from PETA activists, yeah, like activists who against using animal in research involved in this community because yeah, they don't want any animal to become object of research. So there is a screening of this committee. Yeah. Yeah, so in the animal use protocol, there is a description about experiment, number animal been used, justification, what if came up with number, why came up with that numbers, has to be justified by statistic, has to be justified by previous publication, and so on. Description of procedure for cause and pain. This is mostly highlighted what pain that can cause in animal and how they monitor. Yeah. And also about 3R. Yeah, these 3 R's. I will. Yeah. So far, this is 2027 because my boss cannot give me the recent data, so I can use this data. So far in University of Calgary, especially in health science, I highlight here. So we have several committee. This is health science. This is veterinary. We have veterinary faculty, and we have the life science committee. So far, the most protocol in health science, if I calculate, until 1,000 protocols we review. Yeah, either renewals, new submission, modification. Because sometimes some lab propose this number of animal, and then they increase. They have to put modification. Or sometimes the person who's supposed to do surgery is already resigned. So they have to update who's going to do the surgery. Yeah. And everything has to be reviewed. The person who doing surgery they have to be trained. They have to have certificate for surgery. Yeah. Otherwise, they cannot do surgery on animal. Yeah. So this is like in University of Calgary, mostly using mice, some using bird, fish, cattle. We have cattle, rats, and so on. Until cats also. But I think now we don't have cats. Yeah. So yeah, as I told you, review monthly, renewed, yeah, and expired in 4 years. So in 4 years, the researcher has to submit new application again. Even the study is still going on, but they have to submit new application. Yeah, any changes they will include in modification, as I mentioned. Yeah, this is the process, like peer review from veterinary, and then this is like modification through committee, decision, approval. Yeah. And what has been written in protocol has to be implemented. How to make sure? How to ensure the committee will do supervision? Yeah, we come to the lab for post-approval in assurance. So they said that they're gonna inject 1.5 milli. We have to ensure that number, how to be injected, the committee will come to the lab to see. Yeah. If there is deviation from the protocol, we give them love letter from the committee. Yeah, they have to be aligned with the protocol that has been approved. Yeah. Yeah, so ensuring the researcher working within the protocol and for the protection for the animal. And this is the training that I told you before. All the animal facility in Canada must have the institutional animal training. Yeah. From handling, from surgery, from anesthesia, everything. All the student has to be involved in the training. Yeah. Yeah. And the training has to be cover regulation, ethic, and so on. Yeah. Now we come to discussion why this is all important in scientific view. Like my previous question, why this been implemented because of public pressure. But my question now, why we have to pay attention so details about this animal use in research in scientific view? What do you think? It's cheating. I want to from student. Yes, on the back, please.
Thank you for the opportunity
Yeah.
for me to answer. I think the nearest approach is that because we're both, maybe some of the animals is also mammalian, like human, and then maybe for most of the therapeutics or like drugs discovery and things, it's like the final goals is for humans itself. I think the approach through the animals first is like maybe a little bit more less risky, but still it has to be considered to be done very detailed. So I think going through the animals in a more proper and detailed way is the before step before going to the clinical research itself.
Thank you. That's one of the reasons. Yeah, safety in animal will be safety for applicable for the human. Other in scientific point of view, this is more important. The second reason is more important. If the reason itself or the teaching is not for drug discovery or for apply to human, but still scientifically is important to have this animal care control. Yeah, okay, problem with health.
For publication, the high impact journals require you to have met animal ethics standards.
Yeah.
I think Hari can repeat maybe.
Yeah, so yeah, requirement for the international journal to state that your research approved by animal care committee. But do you think why? Why has to be there is a requirement like that? Have you heard about bias, reproducibility? Why this research done in animal giving the different result from other institution using the same animal, using the same procedures? So consistency of the result is important because we use living organism. Yeah. Scientifically has to be consistent reproducible, and far from bias. That's why when we talk about research, yeah, I skip. I will go back to the 3R principle. There are so many factors influence the animal. Stress, like I told you about handling, how to grab the mouse, because I have bad experience myself. Honest story, when I do research, because my research about Fragile X syndrome mouse model is like autism. Fragile X syndrome is the main cause of autism spectrum disorder. The mice shows hyperactivity, you know, like autism patients. Yeah, they're so active, jumpy. Yeah, so it's difficult to catch, to handle. There is a way to handle it, but at that time I use tail to grab the mouse. I use tail. At that time it's not prohibited, but recently there is a research mentioned handling mice, taking mice by the tail, cause stress, and it influenced the result. You can imagine, I made mistakes in the past. That's why sometimes I got inconsistent result. Maybe because the mice is already stressed before we give therapy or we do some behavior test. Yeah. If mice already stressed, how they perform the test? Yeah. So recently, the committee suggests or recommends handling mice by cupping. Yeah, cupping method or using tunnel. Yeah. Just handling. You can imagine. This is about handling. It's not about procedures or just how to handle the mice. Yeah. So we can see in here so many factors like to get science credibility or research reproducibility. Yeah. Avoid stress. Attention to the welfare. So that's why in animal research there are 3 principles: reduce, replace, refine. I start with replace. So if again like cancer research, if there is a method that not using animals, the committee will suggest that method, like using AI, or because so many data outside there that we can utilize through AI or simulator, yeah, especially for teaching, using simulator. The committee will suggest that. If still has to using the animal, there must be justification. For example, teaching. Recently we reviewed protocol using pig as teaching. Yeah. The researcher justified that we can use simulator, and it's been used for teaching using simulator, but the condition still different. So we have to use live animal like pig for this procedure in teaching, and the committee approve it. Yeah. That's replacement. Reduce, we just like I mentioned before, we justify the numbers, how many animals. Yeah, it's not like just roughly. We cannot say just roughly. We have to have precise number by statistical, by previous publication. Yeah. And I give this lecture also, give this seminar in Mula Warman University. That's a good question. I test it to you, like if you can answer it. How about if we test several drugs, agents that never been tested before, and of course when we test it, we have to have controls. Then we require many animals. How we deal with this situation? I got that question in Mula Warman University, government university in Kalimantan or Borneo. Yeah. Any answer? No. Still using mice. They cannot use zebrafish or they cannot use alternative. They must use mice or rat. What can be done? Any answer? No? No, not Proflobby anymore. Using pilot. So pilot is one way to reduce the animal on actual experiment. Yeah. So pilot usually using small number animal to determine the effect, the dose, until what limit, and especially unknown region, what the implication to the animal. There is no publication at all before. The pilot will be recommended by the committee. Preliminary data pilot with small number animals, so they know where they have to stop the dose, how many animals they require. And refine, the last one, refine. Like, who's the student name? Sasa. Caca, yeah. Caca mentioned, if has to use animal, the committee will ensure the refined method, the procedure. Like I mentioned to you, when growing cancer cell on animal, the committee will recommend using analgesic drug, opioid, so the animal won't have pain feeling, because otherwise that also will cause deviation from the result. So that's 3R. And this is the example of experiment using, like in orthopedic, antibody, and also cancer, like I mentioned. And nowadays, as maybe some of you already familiar, using transgenic animal. Because my field is neuroscience. So in neuroscience, we tend to determine circuit of the neurotransmitter pathway signal. Usually, if we use electrical stimulation, all the neurons will activate it on. Actually, we don't want that. We don't want all the neurons will be stimulated. We just want to ensure several pathways, several neurons will activate. Using light stimulation, there is no activation. But with optogenetics, we express some gene that only express on certain neurons. When we stimulate with light on certain wavelength, certain neuron will activate, not all the neuron. Then we can detect the pathway, the cascade, the circuit of neurotransmitter. And we can do it in live animal, like this. So small transplant surgery to embed the light stimulated device, and when we stimulate, that device will stimulate certain neuron and can show certain activity like this. Yeah. See, this is one of implementation of using animal, yeah, modified genetically, and again live animal in vivo experiment. Yeah. And this is my project, drug discovery, but not in cancer, but in neuroscience. And this is traditional way also, using mouse model, autism or Fragile X mouse model. Fragile X is like diabetic. People with diabetic, they don't have insulin, or the insulin cannot regulate properly, so that the doctor give them dose of insulin to be injected to the patient. Similarly, in Fragile X syndrome, they don't express Fragile X protein or FMRP. This protein is important for transcription of the gene, transcription factor of the gene. So when they don't have FMRP protein, certain regulation by that gene transcription will be lack or will be disrupt. That's why this approach is like diabetic patient, we inject FMRP protein. But you know, protein is big, yeah, several kilodalton. How can this size of molecule can penetrate blood-brain barrier? So we came up with the design like how virus penetrate the cells using TAT peptide. So I explore several TAT peptide optimization, which amino acid can really penetrate the blood-brain barrier, and we found it, and then we combine with the peptide, FMRP peptide, and then we inject to this mice model, and we test with immunohistochemistry, and we found there is an expression in this is in Purkinje cells, yeah, in the cerebral brain, yeah. This is just the vehicle. There is no expression, and there is expression. So that the drug penetrate from the tail, from the blood flow to the brain. Because usually people test directly to the brain, yeah. But we can do injection through blood flow to the vein and then come to the brain. But the most important, not this one, this one. So this is a benefit of using traditional way. We can test how the effect of the drug after injection. This is wild type mouse, doctor mice, healthy mice. The trace of their movement. So we put trace. So we measure the movement like this. In the box or in the cage. This is the healthy mice, and this is the Fragile X syndrome mice. Like I have told you, hyperactive, yeah, like this. So it's different. When we inject this peptide, the behavior shows similar to the wild type. There is an improvement on hyperactivity. Yeah. And also when we test the brain activity, this is because Fragile X syndrome also common sign of low capability learning. Oh, sorry, yeah, 5 minutes. We'll be wrap up, sorry. And after injection this peptide, the activity become increase. So learning capability also increased by this peptide. And this is many research on diabetes, liver disease, peripheral nerve damage, obesity, all using animal. So again, I couldn't emphasize more that good animal care will lead to good or impactful research discoveries in many other areas, in life science and healthcare. And that's all. So thank you.